Alright class, so we were talking about aldosterone in the last video. So when aldosterone works on the distal convoluted tubule, it causes sodium reabsorption and water follows sodium. So essentially what we are reabsorbing back is salt water, right, which is isotonic to our body fluid. Okay, now so far we have talked about, okay, this gets reabsorbed back into the system. Glucose getting reabsorbed back into the system, water getting reabsorbed back into the system. But when we say back into the system, where is it going? Are the things being reabsorbed, are they just hanging out in the interstitial fluid, interstitial space? No. Think about it. Do you remember that the efferent arteriole that comes down like this, the efferent arteriole, what does it form? It forms the peritubular capillaries and vasa recta. Do you remember that? So it forms the second capillary bed of the portal system. And this capillary bed that I have drawn, this is called this part, which is on the, actually let me erase this quickly. Ah man, I'm messing this up. Let me do this quickly one more time. Okay, so the efferent arteriole forms a capillary bed over the proximal and distal convoluted tubule, and all the things that are being reabsorbed back from the proximal convoluted tubule gets back into this capillary network, and this capillary network is called peritubular capillaries. All right. Also remember, at the distal convoluted tubule there is tubular secretion, where all the hydrogen ions and excess potassium ions and drug metabolites are dumped into the distal convoluted tubule. So the exchange takes place between the capillary and the tubule. So at this point, I'll just mark this with black. Things are reabsorbed back into the capillary from the proximal convoluted tubule, and here things are dumped into the distal convoluted tubule under normal condition. Only when, listen carefully, only when aldosterone works on the distal convoluted tubule, only then you have sodium and water reabsorption from the distal convoluted tubule. If aldosterone is not there, nothing will be reabsorbed back from the distal convoluted tubule. It is mainly the site for tubular secretion, not reabsorption. Only under the influence of aldosterone, sodium and water will be reabsorbed back from the distal convoluted tubule. All right. Then this capillary forms another network around down here, and the network of capillary that wraps around the nephron loop or the loop of Henle, this part is called vasa recta. Good. So together the peritubular capillaries and the vasa recta forms the second capillary bed of the portal system. So this was the afferent, remember? It forms the first capillary bed called glomerulus. So glomerulus is the first capillary bed. This right here is a portal vessel, efferent arteriole, that breaks up to form the second capillary bed, which is made up of the peritubular capillaries and vasa recta. This will come together and it will form the interlobular vein. All right, class. Hope that's clear. Now we will zoom into the glomerulus. We are zooming in to the glomerulus, and let's look at some structures. Now on top you see a little box, right? And if you look at this box, you do see the nephron, you see the Bowman's capsule, the proximal convoluted tubule, the nephron loop, the distal convoluted tubule, and also the collecting duct. You also see the afferent arteriole, the glomerulus, and the efferent arteriole, right? So we are just zooming into this part, and blowing this up. When you blow this up, what do you see? You see the first capillary bed or the glomerulus. So this capillary network, this is the glomerulus. Surrounding the glomerulus, This part is called Bowman's capsule. This is the Bowman's capsule, right? And inside the Bowman's capsule, this space is called glomerular space. Glomerular space. The Bowman's capsule leads to this part. This is part of the proximal convoluted tubule. We also see the afferent arteriole with a larger diameter. This is afferent. And the efferent arteriole with a smaller diameter. Okay, another thing to remember, and we did talk about this during the blood vessel lecture. The glomerulus has fenestrated capillaries, meaning the capillaries have holes, and these holes help to filter material out of the blood. So there are holes on the capillaries. So the glomerular capillaries are fenestrated. All right. Okay. So this is just another representation. I just want you to see that this is the first capillary bed, glomerulus, and then if you notice the proximal convoluted tubule and the distal convoluted tubule, there is this pink colored capillary net that is covering them up, and this capillary net is called the peritubular capillary, and then along the loop of Henle there is the second capillary bed called vasa recta. Okay, they come together, the peritubular capillaries and the vasa recta come together to form the interlobular vein. This is the interlobular vein right there. Okay. Okay, so now let's look at the PowerPoint. So we know that the nephrons are the functional unit of the kidney. There are about 1.25 million nephrons per kidney. We talked about this before. And the nephrons are the little structures that actually filter blood to produce urine. Urine formation takes place through three processes. We have glomerular filtration, tubular reabsorption, and tubular secretion. And finally, the final product that comes out of the collecting duct, that's called urine, which is stored in the, which is carried out of the kidney by the ureter and then stored in the urinary bladder for some time before it can be excreted out of the body. So right here, we see the two types of nephrons present in our body. The first one is called cortical nephron on the left hand side. And the cortical nephron, as you see, they lie mostly in the cortex. So this part is the cortex and this part is the medulla. So the cortical nephron lies mostly in the cortex. Only a tiny bit of the loop of Henle can actually dip into the medulla. The second type is juxtamedullary nephrons, and in this case, the part of the proximal and distal convoluted tubules are in the cortex. It has long nephron loop, and the nephron loop mostly lies in the medulla. So that's the difference. That's the anatomical difference between cortical nephrons and juxtamedullary nephrons. Okay, looking into the anatomy of the nephron. The anatomy of the nephron is made up of, we will talk about the renal corpuscle, proximal convoluted tubule, nephron loop, and distal convoluted tubule. So let's take a look here. All right. So here, what do we see? Let's go over this diagram. So this vessel right here is the afferent arteriole. This is the glomerulus right there. I should use a different color. And this is the efferent arteriole. Surrounding the glomerulus, this horseshoe-shaped structure is called the Bowman's capsule. The Bowman's capsule leads to the proximal convoluted tubule that leads to the loop of Henle, descending limb, ascending limb, that leads to the distal convoluted tubule, which ultimately joins the collecting duct. Okay. What else? Very, very important. If you think of this entire thing, which includes the glomerulus and the green colored Bowman's capsule together. So glomerulus plus Bowman's capsule or glomerular capsule, it's the same thing. Together, it is called renal corpuscle. So renal corpuscle is, when we say renal corpuscle, we are actually thinking of glomerulus plus the glomerular capsule or Bowman's capsule. So the whole thing here. All right, another thing I want you to notice. Look at the efferent arteriole. Look at the efferent arteriole. Do you see how the efferent arteriole branches to form peritubular capillaries and vasa recta? Do you see that? And then this capillary, this is a second capillary bed, right? The first one was glomerulus. And then these capillaries come together to form the this part will be called the interlobular vein. Hope that's clear. All right. So again, zooming into the renal corpuscle, and okay. So if you think we are looking into the renal corpuscle, and what do we see here? If you look into the renal corpuscle, you have a tubular pole. This pole where the proximal convoluted tubule starts, that's the tubular pole. And this pole is called the vascular pole where the blood enters. Do you see how the blood comes in to the glomerulus and then leaves the glomerulus? That's the vascular pole. I also want you to notice here. Do you see there is this tubular structure here? This is actually part of the distal convoluted tubule that loops back to kiss the afferent arteriole. If I go back, class, let's look here. Look at this particular nephron. Anatomically, it looks slightly different from what we drew out. But what I want you to notice is that this is the Bowman's capsule, proximal convoluted tubule, loop of Henle, distal convoluted tubule. Now see, the distal convoluted tubule loops back to kiss the afferent arteriole. Do you see this junction? So the distal convoluted tubule is kissing the afferent arteriole, and this kiss is really important. It has very important anatomical significance, which we will focus later. But I do want you to know that the distal convoluted tubule can loop back and it can touch the afferent arteriole. All right. Okay, so vascular pole, tubular pole. Then if you think of the Bowman's capsule or the glomerular capsule, it is actually made up of two layers of cells. The outer layer is called the parietal layer, like this layer right here, and these are simple squamous epithelial cells. That's the parietal layer. The visceral layer is made up of specialized octopus looking cells. These are called podocytes. So do you see these orange colored, I'll just highlight one. Do you see these orange colored cells? These cells are called podocytes. Remember, podocytes are found on the visceral layer of the glomerular capsule, and these cells have some special features. What they do is they just cling to the glomerular capillaries. They are tightly attached like a leech. They are tightly attached to these glomerular capillaries, and they form something called filtration slit that we will talk in a minute. But these are podocytes. So in your exam, you might get a question like, which cells are, okay, if I say, name the cells that are present, that make up the visceral layer of the, glomerular capsule and cling to the surface of the glomerular capillaries. Your answer should be podocytes. Okay.